TICA's webinar series. The recording's in progress. I'll just jump a little bit ahead because we record this webinar series and we upload them to our YouTube channel. And I'll send a link in the chat to the YouTube channel. And so if you prefer not to be recorded, just mute yourself and turn off your video. And as a quick reminder, this webinar series is on the first or on the second Monday of every month at 11 a.m. Pacific time. And if you're not a TICA member yet, but you're interested in joining, I'll drop the registration link also in the Zoom chat after the introductions. So today we have a really exciting lineup of speakers and a really diverse array of topics. We'll be going from the biogeography of the haploid with Carme to talking about ancient hybridization with Paige. And then we'll also finish with a nice flash talk by Isaac, who will introduce us to a tiny new comp. And so we'll have three talks, and the first two will be about 15 to 20 minutes, and we'll have time for Q&A and discussion after each of the talks. And then we'll finish with the flash talk. And if there's any extra time, we can open it up for discussion and chat. And just a reminder that this webinar is recorded. And we'll start with Carme Blanca Gavilao, who will be joining us from Barcelona. She'll be presenting a bit of her cool PhD work on the haploid. So I'm going to hand it over to Carme. Carme, are you able to share your screen?
Yeah, let me try. Can you see it now?
Yes, you're in the like the PowerPoint view. So I think you just perfect. Looks great. All right, go for it, Carme.
Wait, does it work if I change the slide?
Yes.
Okay, right, perfect. Okay, so hi everybody. I would like to thank you all for being here despite the time zone differences. And of course, I would also like to thank the organizers of the TICA Talks for giving me the chance to share our results here today. As you can see from the title, we will be discussing the drivers of the diversification of the haploid in mountain systems. Why are we focusing on high elevation ecosystems? Well, despite their relatively small area, they host many biodiversity hotspots and high number of endemic species. And this is linked to their sharp environmental gradients that offer diverse habitats. And all these factors make them ideal and natural laboratories for studying evolutionary processes. Another question is, what drives diversification in these regions? Previous works have explored this matter and have provided several factors that might have had a high impact on high elevation biodiversity. On one hand, we have biotic factors. These are mostly related to the physical characteristics of mountain systems. Present-day factors include elevation, geology, and climate, but we also must consider the epochime events such as orogeny and climatic oscillations, which significantly impacted lineage colonization and survival on a longer time period. A specific process that has repeatedly been linked to mountain biodiversity is known as flickering connectivity and is associated mostly to the glacial cycles of the Pleistocene. Basically, what it says is that climatic fluctuations isolated and reconnected populations several times, impacting gene flow and diversification rates. On the other hand, we have biotic factors related to species interactions and intrinsic lineage characteristics. It's worth mentioning key innovations, which are new traits that allow lineages to exploit previously inaccessible niches. And this might be crucial to surviving in the extreme conditions of high elevation habitats. Most studied innovations are related to life forms. Other mechanisms are, for example, long-distance dispersal of pre-adapted lineages And just to give you an example, in this case, temperate lineages already adapted to these alpine environments will have a competitive advantage over tropical lowland species when trying to colonize high mountains in the tropics. And of course, all these mechanisms didn't happen in isolation, and they had they had an additive effect, and the factors that interacted and the prevailing one depend on the mountain system that we're studying and also the lineage of interest. Finally, we get to what we were waiting for. What is the hub plate and why is it relevant? Well, as you all know, in our favorite plant family is very diverse, but this is especially true in high elevation systems. And in fact, it's one of the most species-rich plant families in the highest spots of the world. And this brings us to the hub plate, which stands for Helichrysum, Anaphalis, and Pseudoanaphallium, but it also includes smaller genera and constitutes approximately around 800 species. In this map, I roughly indicate the distribution of each genus, and just looking at it, we can already tell that we can consider the hub plate a megadiverse pan-mountain lineage, ideal for studying high elevation ecosystems. But there are also other interesting things. We observed some patterns in what we could consider key traits. For example, high elevation species usually have wide involucral bracts, and life forms vary depending on habitat type. These observations made us wonder, what's behind the clade's evolutionary success in high elevations? Is it its inherent adaptability or the acquisition of apparently key innovations? So our first goal here was to provide an updated phylogeny on the hub clade because it was missing until now. And with it, we wanted to test several hypotheses that we formulated, taking into account our previous works on the tropical Africa and Malagasy mountain lineages. First, we suspect that the hub clade evolved in a pattern of nested radiations in which multiple interconnected radiations led to a massive radiation. Second, the current distribution and diversity of the clade results from the combination of geographic speciation and ecological speciation involving long-distance dispersal and in situ diversification. Next, we want to test whether diversification rates were higher in higher elevations, potentially due to the acquisition of key innovations, although we suspect that broad-scale climate oscillations had a major impact. And last, we also want to test for parallel adaptations to similar microhabitats across the clade's distribution. So to test this hypothesis, we sampled around 62% of the hub clade, including more than 500 species. On the left, you have a pie chart showing the proportion of species per genus in the whole hub clade. And to the right, the brighter colors indicate the percentage of species that we sampled for this study. Then we perform family-specific target capture. We process the samples. We build multiple phylogenies using different approaches, and we dated the concatenated phylogeny. With this, we perform ancestral range inference and biogeographic stochastic mapping. To do so, we defined 6 main biogeographic areas that we further divided into mountain or lowland areas. And with this, we wanted to infer the frequency and directionality of dispersals across space and time, but also assess the contribution of elevational and latitudinal transitions to mountain biodiversity. We also reconstructed the evolutionary history of 2 functional traits, bright color and life form. and to ecological preferences, habitat, and elevational belt. Finally, we get to see some images. Here you have examples of the 7 life forms that we defined and the 2 main bright colors that we find in the hub plate. All of them are Helichrysum species, except the first one that's Pseudoanophallium. And just from looking at it, you can already get the idea that this clade is very diverse. And here you can see how different species look like in the 5 habitat types that we have defined for this study. Again, all of them are Helichrysum species, except the 2 middle ones. At the top, we have Anophalis, and at the bottom, we have a Chiroclinia. Okay. Finally, we studied the macroevolutionary dynamics by testing whether diversification rates varied with time, climate, species diversity, or the acquisition of specific traits. And we also assessed for a correlated evolution between pairs of functional traits and ecological preferences. Moving on to the results, we confirmed that the clade originated in Southern Africa in the mid to late Miocene. It quickly colonized the Southern African mountains, which became the initial center of diversification and main source of lineages. During the Pliocene, it radiated in the tropical African mountains and expanded beyond Africa. But it wasn't until the Pleistocene when several parallel mountain radiations took place. The arrows on the phylogeny to the right indicate the multiple parallel radiations that we detected. This plot might give you a clear idea of what I just explained. Initially, the hub clade was mostly a lowland adapted lineage. However, by the end of the Miocene, high elevation species became dominant. And interestingly, our data strongly suggest a fourfold increase in net diversification rates coinciding with these parallel mountain radiations that happened around 1 million years ago. If we now look at the frequency and directionality of dispersed cells, first, we see that downward dispersed cells from high elevations to lower elevations prevail, suggesting high permeability across elevational belts. And the second most common type of movement is long-distance dispersal between high elevation regions. And this would back up the nested radiation hypothesis. If we now look at trait evolution patterns, on one hand, it seems that white bracts and the colonization of high elevations did not drive higher diversification rates. Instead, unknown factors are responsible of it. And on the other hand, functional traits and ecological preferences evolved multiple times independently across the clade's phylogeny, suggesting that trait flexibility has been a key element in the evolutionary history of the hub clade. Lastly, we did find significant associations between the functional traits and the ecological preferences, suggesting high adaptability and indeed convergent evolution in the hub clade. Here you have some examples of traits and habitats that usually go together. And just to name a few, therophytes are highly correlated with being lowland species, and cushion-like forms prevail in high elevation rocky habitats. And to summarize, we do provide an updated, comprehensive, and well-resolved hub clade phylogeny, including taxa from across the clade's distribution and most taxonomic groups. And our findings suggest that the hub clade's evolutionary history is dynamic and complex, and that its success stems from adopting flexible strategies that involves the interplay of multiple factors. Specifically, we propose a kind of a multi-stage process that involves long-distance dispersal, which repeatedly connected geographically distant mountain regions, driving geographic speciation. And at a more recent and local scale, it seems that the cycles of isolation and reconnection that took place during the Pleistocene also contributed to geographic speciation. And after this, well, this was followed by local scale speciation. This was likely facilitated by the clade's high adaptability and trade flexibility that we talked about before. And all this led to ecological speciation. And all these processes together resulted in these multiple parallel mountain radiations that took place across the clade's distribution. And to finish, I would like to mention this, although we didn't explore it in this work. We suspect that allopolyploid speciation might have played a major role in the massive radiations that took place in Eurasia and America. And this is a really exciting matter, but of course, tackling this issue requires a different approach, and we save this for future works. So this is all. Thank you all for being here. Of course, a huge thank to all people that contributed to this work, and I will be happy to take any question.
Awesome. Thank you so much, Carme, for that beautiful and talk and really impressive work. I want to open it up for any questions, so feel free to unmute yourself if you're comfortable doing so and ask, or drop a question in the chat.
Thank you for your wonderful talk, Carmen. I had a question about the how the cushion plants were found both in rocky outcrops and in mountain areas. Were the rock outcrops typically at higher elevations, or were they rock outcrops that might be found in other types of environments?
Yeah, this is a good point. These rocky habitats are also found in lowlands, but we found correlations between both. So we can deduce, or we can infer that it usually happens in high elevations. It's just what we guess.
Awesome. Thanks, Carme. If there's any other questions that come up, and we'll have time at the end, hopefully, to continue the discussion. But thanks for the great talk. Next, we're going to move on to Paige, our next speaker, and she's joining us from the University of Memphis, and she's exploring the big topic of the evolutionary complexity within Asteraceae. Paige, I don't think we can hear you.
You can't hear me?
Now we can.
Okay, great. Okay. I'm on like a different setup and I actually haven't tested the sound, so I was like, oh, that'll be bad. Yeah.
Just to interrupt really quickly, I don't think we can see your screen.
Okay, just a minute.
We can see for a minute oh wait. Yeah, we can see the PowerPoint mode, and
okay.
I think the issue is when you go to full presenter. So maybe try now.
Okay. Okay, are you seeing it? I might need to move it to a different screen.
No, it's perfect. All right.
Oh, it's good.
Thanks, Paige.
Okay, fantastic. All right. Yeah, thank you, Carme. That was very interesting. And yeah, I'm glad you brought up the allopolyploidization at the end. So that's a good segue into what I'll be talking about today. So first of all, thanks for having me here at these TICA talks. My name is Paige Ellisted. I'm a postdoctoral researcher at the University of Memphis, and I've been working in Dr. Jennifer Mandel's lab for about a year and a half now. So initially I was excited to join Dr. Mandel's lab because of this extreme diversity within Asteraceae. And I thought that this is going to be a great study system to address some of the most difficult biological and evolutionary questions. And so all my work so far has been focusing on using Asteraceae as a model, mostly at the family level. It's been great learning more about its complexity at other levels, like tribal levels, genus, and then down to the species level. So today I wanted to share with you some of the research that we've been conducting to explore the complex processes that have influenced Asteraceae diversity. And specifically, I'll be focusing on our efforts to try to identify ancient hybridization within the family. Okay, so as we all know, Asteraceae is a hyperdiverse family of flowering plants that inhabits every continent on the globe. With up to 35,000 species, its species richness far surpasses that of related families in the order Asterales. As a major factor forming this diversity, its evolutionary history is characterized by a series of explosive radiation events, which are influenced by global climatic changes. So Asteraceae and its sister family Cheliceraceae originated around 83 million years ago in southern South America in the late Cretaceous. And then it expanded into northern South America. Then during the early Eocene, it went through a major radiation event into Africa, Asia, and North America. And in the mid-Eocene, it radiated from South Africa, Southern Africa, into Northern Africa, Europe, and Australia. And then its diversification rates peaked around 23 million years ago during the Miocene, where it radiated back to North America, Central America, and South America. This series of radiations has greatly contributed to the evolutionary complexity of the family and has been evidenced by diversification rate shifts along certain nodes in the phylogeny, along with evidence for whole genome duplication, occurring in multiple ancient and then some more recent lineages as well. And then on top of that, there is support for the occurrence of reticulation events within some lineages. So here are a few, just as examples, where some hypotheses have been proposed for hybridization within the tribes Perdii and Senecio, and then also hybridization has been shown within the genus Helianthus. There are many more examples, and if anybody wants to send us more, that would be great. So these factors, combined with the occurrence of incomplete lineage sorting, has contributed to some of the phylogenetic discordance that often comes up within this family. So this figure from Palisezi et al. Exemplifies some of the phylogenetic discordance that is commonly found in Asteraceae. So first of all, we have instances of nuclear and plastid discordance, as shown by the blue circles on the phylogeny. And then we also have regions where there is substantial gene tree conflict, and that's shown by the orange circles. So for this study, we wanted to use all of these nodes, these nodes of phylogenetic discordance as focal points to try to better understand the complex evolutionary processes that have influenced Asteraceae's diversity. And we did that by accounting for incomplete lineage sorting during our phylogenetic reconstruction, and then by identifying major events like whole genome duplication and or reticulation. So for this, we wanted to have sampling as comprehensive as possible. So we were able to incorporate sequencing data from 267 taxa, which encompassed 47 tribes in Asteraceae. Then we had one taxon from the Asteraceae sister lineage Cheliceraceae, and two taxa from the related family Goodeniaceae. We primarily used target-enriched HiBSeq data, which was publicly available, but we also generated additional HiBSeq data for taxa from the tribes Fedii and Polymnii. And then for a subset of our data, or for our taxa, we incorporated publicly available transcriptome data, and that was really focusing on taxa of the understudied South American lineages. And we also generated our own transcriptome data for 22 taxa. So to account for the unique evolutionary history of genes, we used a coalescent-based method for phylogenetic reconstruction. And then we compared that to the more commonly reported concatenated tree. After that, we compared nuclear and chloroplast trees to identify cytonuclear discordance and possible hybridization. And then we assessed synonymous substitution rates to identify major duplication events. Then we generated phylogenetic networks from subsetted groups to identify ancient reticulation events. So after generating a coalescent-based phylogeny, we compared it to the concatenated phylogeny, and we found some discordance between the two. So in this figure, we have the concatenated phylogeny by tribe on the left-hand side and the coalescent phylogeny on the right-hand side. And based on this visual, this tangogram, you can see where there are quite a few places where the tribal relationships are not the same. And also just to point out, even though some of the connecting lines are straight between the two trees, these relations still might differ. So I'll show some examples of some of these relationships that have differed. So here are it's the base of the tree, expanded to show the taxa that we used. And I just wanted to point out a few of these major differences. First of all, the most striking incongruence between these two trees is the placement of the Cheliceraceae taxon, which we use Gammocarpa macrocephala for this. So in the concatenated tree, it's sister to Asteraceae, while in the coalescent tree, it's actually nested within Asteraceae, although it does stem from a node with low support. And this is actually not the first time this relationship has been shown. It's also been shown in both of these papers. But also in these papers, again, both topologies reported low support for this relationship. And then further throughout the phylogeny, we are seeing different placements with the tribe Famatinanthe, Nisalvii, Wunderlicki, and Hecastically. And there are quite a few more differences, but I'm not going to go over them all. So next, to identify cytonuclear discordance and possible signatures of hybridization, we then generated a chloroplast phylogeny by mapping the off-target reads to related chloroplast genomes. So you can see here with the concatenated tree on the left and the chloroplast tree on the right, there are some substantial changes in topology and relationships. This may represent accurate evolutionary histories of the plastome. However, the topology may also be influenced by the fact that not all of these samples had complete plastome sequences that were able to be mapped. And it could also be due to the fact that there were a couple of rearrangements detected among the samples. So these rearrangements could have decreased the quality of the alignments and biased phylogenetic inferences. Because we're able to construct complete plastomes from some of the samples, actually 22 of them, we were able to identify these rearrangements by looking at collinearity. And from our complete plastomes, we found 4 different plastome structures, or general plastome structures. One which included most of Asteraceae. However, the plastome of Gerbera from Mutici exhibited a small inversion. The plastome of Gammocarpa macrocephala, which is from the sister family Cheliceraceae, exhibited a large inversion. And then Schlechtendalia from tribe Barnardizzi exhibited also a large inversion, but one that was different from Cheliceraceae and the rest of Asteraceae. So it was unique. So we then tried to compare the past signatures of whole genome duplication by assessing the KS values for all taxa that we had transcriptome data for. We used WGDV2 to identify peaks within the KS distribution data, and that allowed us to determine the relative timing of each past major duplication event. But due to the fact that that substantial gene fractionation may have occurred, especially coming from older events, and then also that fractionation rates may vary by taxon, we weren't able to directly compare our results by taxa, but we were able to make some hypotheses on which events were shared or not based on their similarity in KS peak values. So for example, here on the left, we have AC Carfa from the family Caliceraceae and Arnoldoa from the tribe Barnardisi. And they have their KS peaks at 0.47 and 0.35. So these peaks are close enough, meaning that it is possible that they could be indicating the same shared event, although it's still a little open. And then contrastingly, we can see for Amelia, it has a peak around 0.94, but also an additional peak around 0.14. So it's a much lower KS value. So that likely indicates that it had a much more recent whole genome duplication event. So overall, when we plotted all of our KS peaks, every peak that was detected by this program, we plotted them by tribe. We found really varied results. For example, some tribes like Senecionii and Nephalii had evidence of more recent whole genome duplication in some taxa, but not others. So this could indicate a more lineage-specific duplication event. And since we were not able to directly compare KS rates among our species, we found that the placement of most whole genome duplication events, or not most, but a lot of them were pretty ambiguous. It was really hard to pinpoint where in the phylogeny it should be. So for our final suite of analyses, we wanted to narrow down our hypotheses on whole genome duplications by seeing if some of these signatures we found might have actually resulted from hybridization. To do this, we focused on the early diverging lineages of Asteraceae and assessed reticulation using a program called PhyloNetworks. And since this program is computationally pretty intense, we had to subset our sampling into groups of like 10 to 16 taxa. So we made 6 groups here, and this figure shows how we separated our groups. And the program helps to identify the optimal number of hybridization events, and then also the taxa involved in these hybridization events. So for our first 2 subgroups, we found support for hybrid edges involved in the formation of species or a species within the tribe Barnardisi, the tribe Muticii, Onoceridii, and Nasalvii. And these are species-specific analyses, so I can't quite say the whole tribe, but I'm just generalizing here. And then from our second 2 analyses, we found that hybrid networks formed species from the tribe Purdii, Muticii again, Dichomii, Oldenburgii, and Tarquinanthii. And then from our last 2 analyses, which focused on tribes adjacent to Barnardisi and then tribes along that whole section that I showed in the first one of these figures, we identified how hybridization helping to form species from Barnardisi, Hyalidii, Muticii, again, Wunderlichii, Cardui, Oldenburgii, Dichomii, and Purdii. So when we plotted these back on our overall phylogeny, based on taxa that was and wasn't included in the subgroup sampling, we found these nodes where many hybridizations likely or might have originated. So I just circled these nodes in red. So these are possibly indicate reticulation hotspots in Asteraceae's evolutionary history. So combining all of our data together, we found that although many of these results did not allow us to completely pinpoint specific events, we definitely found signatures of reticulation, quite a bit of it, and whole genome duplication, a lot of that too. And particularly among lineages and nodes shown by these purple stars that I put in this figure. And you can see these do often coincide with many of the previous hypotheses of whole genome duplication and also the diversification rate upshifts. In conclusion, we found that overall reticulation has played a major role in the diversification of Asteraceae, but there are some very real biological and bioinformatic challenges to accurately detecting these events. First being that due to extinction, we'll never have a full picture of Asteracea's evolutionary landscape. And although we can try to have a comprehensive sampling, there still might be key missed taxa, which help to support or refute the placement of duplication and or reticulation events. Then also the quality and completeness of our genomic data sets might also be influencing our results and therefore our inferences from them. And so there's still obviously quite a bit of work to do to disentangle these complex events, but some of these challenges may be overcome through the incorporation of more high-resolution genomic data, especially from many of these understudied lineages. And as a way to increase transparency and reproducibility of all the analyses that we conducted for this project, we have made a bioinformatics tutorial describing all of these analyses. So it's hosted on Erica Moore Pollard's GitHub. So if you'd like, you can take a look at it and let us know what you think of it. It goes over how to install programs, run analyses, and down to creating visualizations. So hopefully it can be helpful to somebody out there. And yeah, if you have any questions about it, you can contact either of us. So, and with that, I'd like to thank everyone who has helped with this project with acquiring samples and generating genomic data. And then I'd especially like to thank all the co-authors who graciously included me in this collaboration. And with that, I will take questions.
Paige, thank you so much for that presentation. This is a really impressive body of work, and I think it opens up many, exciting new questions, especially around ancient hybridization in the family. And also big kudos for the bioinformatics tutorial. It will definitely be helpful for lots of people, and this trend towards open science is really crucial. So is there any questions from the group? I'm opening it up now. Looks like we have one in the chat from Ed. I'm going to read it out loud for you, Paige. It said, how does phylogenetic reticulation affect estimates of clade divergence times?
And so we can place them on previous phylogenies. Like I said, a lot of these analyses kind of have to be taken with a grain of salt due to sampling and I guess also how you get your divergence times. There's so many like hypotheses on hypotheses that makes it kind of hard to say exactly where the reticulation events take place. We wanted to try to kind of figure out a way to do that with confidence, but we've just been placing them back on previous phylogenies with divergence times. So that's how we've been handling it.
Any other questions? Thanks, Paige. We'll have again time for open discussion, and it was a really excellent talk.
Thank you.
We'll finish with a little flash talk on little comps from Isaac, and I'll give you the floor. I think you're muted, Isaac. One second. Perfect, looking good.
Okay. You can see the slides?
We can see and hear you, so go for
Okay,
it.
great. Okay, so just if my internet's not great, so Lizzie, if you would please just stop me if you can't hear me, okay? So thank you, everybody, and thank you especially to Lizzie and the rest of the organizers of the TICA Talks for allowing me to do this flash talk. So my name is Isaac Lichtermark. I'm a postdoctoral researcher at the California Academy of Sciences in San Francisco, California. and I'm also a member of the TICA Talk organizing committee. Um, at some point, I'll probably be able to introduce myself in more detail. I study the Helianthi Alliance more broadly. But the goal for this flash talk is to introduce a new friend, the woolly devil. This is a new genus and species of Compositae that I was lucky enough to work with some co-authors on describing earlier this year. And it's just a really cool plant. It's a small composite that is completely covered in really dense, fuzzy wool. It has few, usually three or two elongated ray florets that are white with maroon lines act as kind of runway for pollinators to find the inconspicuous inflorescence of this plant. And it's an ephemeral annual, a small plant that grows in the backcountry of the Chihuahuan Desert in western Texas. The story of this plant, it's a relatively recent discovery. So last March, these two botanists, Deb Manley and Kathy Hoyt, were hiking through the backcountry of Big Bend National Park, and they found this really weird looking plant, that was looking at them, and they thought this must be, another really difficult to recognize small composite. So they took pictures and they posted it to iNaturalist, and immediately it caused a huge stir, and people were emailing each other and saying, "What is this really weird comp?" And they got a couple of the really knowledgeable local botanists in West Texas involved, including Michael Powell and Kelsey Wogan. And they worked together with people from the park. They got a permit to collect the plant, and they actually sent material to me at the California Academy of Sciences in May. And I had just started working with this really great student, Kaylee Peralta, through an NSF RU program. And Kaylee was really excited about doing some sequencing and some SEM. And we got this envelope with the woolly devil inside it. We opened it up and found these little dried woolly balls. And basically, we just dropped everything we were doing, and we were like, we have to study this plant. It's so cool. So we sequenced ITS of the woolly devil, and we plugged it into existing kind of data for the Helianthi Alliance. We found really strong support that it's a member of the Helenii tribe, so the sneezeweed and blanket flower tribe, and specifically part of the Tetranurinae subtribe. And that made a lot of sense just based on kind of macro morphology. But one thing we found is that it did not fall cleanly into any of the well-recognized Tetranurinae genera. It was kind of on this unique evolutionary branch, sister to Silastrophe, the woolly paper flowers. And that, was kind of surprising. And there were a couple bits of evidence that kind of back up that result, the uniqueness of this plant, which suggested it needed to be classified not only as a new species, but as a new genus of Compositae. And one of those, here it is on this unique branch here. One of those is that Michael Powell has generated some information to support a close relationship with Silastrophe based on meiotic chromosome counts. And another is just if you look at the morphology of this group, here's examples of plants in the Helenii tribe, which all look very beautiful. And then there's the woolly devil, which looks like the director Tim Burton kind of took a turn creating a composite in the Helenii tribe. So it really stands out as a very unique and interesting plant. And when we published this, we published it as a new genus and species. And Deb Manley, who discovered the plant, actually chose the name. She decided to name it Ovicula, uh, which means little sheep, because of its woolly appearance and also because it has these little ray florets that kind of look like horns. So, I like this headline here in Scientific American. Part of, the big enthusiasm about this plant is that I think it showed that sunflowers, people recognize the classic kind of domesticated sunflower, but that sunflowers can also be super unique and call our attention to the strange ways in which plants adapt to ecosystems. And the woolly devil has some good examples of that. One of those is its fuzzy wool, which shows up in a lot of different types of composites, including some of the ones we were hearing about from Carme earlier in the Nephalii tribe. And we looked closer at those hairs using, microscopes and SEM, and we found that these are actually, the kind of helical hairs that show up in a lot of those other diverse types of composites, and that are predicted to play a role in kind of sunscreen to protect the plant from UV light or create a boundary layer from desiccation. So recently we've thought about testing that to see if these plants absorb UV light by shining a UV flashlight on the woolly devil. And we find that, yeah, actually the foliage does absorb UV light, but we were pretty surprised to see that the ray florets do not. They reflect it, which is kind of interesting because it suggests pollinators, which often see in UV radiation, might be attracted to these rays. They may, be a lot more conspicuous to those insects than they are to us. And also, the woolly devil could be a really great Halloween costume. Um, these ray florets themselves are pretty unique when you look at them under a microscope. So here's the ray floret corolla detached from the fruit, and it has a lot of those woolly hairs along the corolla throat, and then on the abaxial surface of the strap-shaped corolla are these unique short stipitate glands, which show up in a lot of different types of Asteraceae and in general tend to be associated with sesquiterpene lactone synthesis. So they may be producing kind of unique chemicals that protect the plant. The fruit itself is also beautiful, and what we were able to see with SEM that the scale-like pappus elements have this really beautiful kind of pleated structure, and they come to a kind of tapering tip. And the hairs that are found on the fruit are very different from the hairs that are found on the foliage. They are these kind of classic examples of fruit hairs that you see in all different types of compositae that are fork-shaped and that have been called Zwillingshaar. So far, the woolly devil has only been documented in a very narrow range, which is a bit concerning. We were only able to work on it from examples from 3 small subpopulations in the backcountry of this national park. But since the publication of the plant, a new population has shown up on iNaturalist. The plant was actually observed in 2019 by somebody. They had the photos on their computer and then they joined iNaturalist and shared these. So it actually represents a pretty significant range extension for the woolly devil, about 80 miles to the east in the small town of Sanderson. But even if we wanted to, we couldn't go and see these plants today because the entire region is under an exceptional drought. Even things like creosote and cacti that are relatively resilient to drought are dying in this region right now. And it's concerning because these are conditions that are predicted to become more common because of climate change. And so all of these plants like the woolly devil that survive on the good times, years where there is a good amount of rain, are predicted to become less common over time. And so because of that, we argued that the woolly devil is probably a very vulnerable species and one that we need to go out and learn everything we can while we, still have the opportunity to do so. Um, so as soon as rain falls in this region, there's going to be botanists kind of, looking everywhere for the woolly devil, and hopefully we can find more populations. Uh, Michael Powell has also been successful in domesticating the woolly devil to some extent. Here's an example of a plant growing in the window of his kitchen. And we've also been lucky enough so far to work with Vic Albert at CUNY to take some material from the domesticated woolly devil and begin to generate whole genome sequence data. So as new populations are discovered, we could begin to understand their potential for, preserving the genetic diversity of the woolly devil in the long term. So that's the extent of my flash talk about the woolly devil, just introducing this really cool new comp, and just mentioning that it's an example of a new plant species and genus discovered in a national park in the U.S., which I think has called a lot of attention to how little we know about our co-inhabitants on the Earth, but especially in places like deserts. Um, and also how composites are not only really diverse, but also very weird and strange plants that can inspire people to understand, the possibilities for evolution in these kind of extreme climates. So thank you for letting me present today, and thank you to TICA and to everyone who supported this research, especially my co-authors on the woolly devil. And I'll be happy to take any questions.
Thanks, Isaac. This new species has like everything going on in it with like the citizen science and, the sobering, I think, trend of describing new species that are already threatened and such amazing science outreach and communication with this plant. So very cool uh, presentation and exciting new description. Is
Thank
there
you.
is there any questions for Isaac? I might jump in, use my hosting privileges to jump to the front of the line. Um, I guess I wonder, so the first observations were the iNaturalist, citizen science. Has anyone looked back in like a herbaria that specialize in this region to see if there's kind of any earlier collections hidden in like index sections, like, or are those, you think those iNaturalist observations are truly the first recording of this new species?
Yeah, that's a great question. So, as part of this research, one of the things we did was scour some herbarium collections. Obviously, it would be very difficult to say you've done that in kind of like a comprehensive way, but we looked in some of the major collections for this region, and this was a binational team, and so Arturo Castro, who was part of the team Woolly that worked on this plant, was able to scour collections in northern Mexico that may have had, some examples of the woolly devil, like you said, kicking around. It's possible that some of them, if they were collected after they were dried up and those ray florets were missing, might have been filed as Nephalidae in some cases because they look very similar without the ray florets. So maybe they will turn up. And in terms of iNaturalist, there's a really great community on there, and a lot of people kind of looking around. And there were some possible observations around, and then there's some people who claim they plant, but they just forgot to take photographs. But those are just rumors. We don't really have any proof about that.
Awesome, thank you. Are you getting some questions in the chat? One question is, any idea what the Sipsily look like?
Oh, yeah, I can definitely share the screen again and show the picture um, that I shared with the Sipsula. So bear with me here. Here's just a photo I threw in here of the fruit. This is from a disc floret. Can you see it? Okay, yeah. Okay. Am I sharing the correct slide?
You are.
Yeah, okay, perfect. So this is having seen this and seen the plant itself, especially those ray florets, I think there were a lot of people that already were forming in their mind a hypothesis that this might have been a relative of the Hellenii tribe. And a lot of people probably guessing that it was a member of the subtribe Tetranurinae, because members of this tribe often have this shape of cypsela, and they often have pappus elements that are modified into these kind of tapering hyaline scales. And a lot of the members of that subtribe, or not a lot, but many of them have ray florets that have those maroon lines or have kind of dark lines in them that are especially apparent when you collect the plants and then dry them onto an herbarium sheet. So just based on the shape of the cypsela and some of those other macromorphological characteristics, there were many people, even people on iNaturalist, who threw out hypotheses that were correct about the relationships of this plant. But I just highlighted here, some of the unique things that came out of doing scanning electron microscopy on this plant, including the pleated structure of these scales, which might have kind of some more developmental information packed in that might help to, kind of sort out information. Try. We also took photographs of other traits like pollen, but those tend to be pretty homogeneous across these groups. I'm happy to answer any specific questions about the fruit you might have had.
There was a follow-up question as well. Maybe when we're talking about morphologies, is there any hairs to help them anchor in the soil if and when it rains?
But that's a great, I'm not sure, but that's a really great hypothesis for, potentially why these fork tip hairs are so common in Asteraceae. Maybe they do play a role in kind of anchoring the cypsela in place. I've never, encountered that before, but it's a great idea.
Any other questions for Isaac? No, I think that's it. Thank you so much, Isaac, for
All
this
right.
talk, and also to Carmen and Paige. This was for the webinar, such a really diverse and beautiful array of talks and methods and systems. So thank you so much to all our speakers today. And with that, I'll say goodbye and thank you for coming. Our next one is next month, and we'll send out a another flyer with talks a few weeks ahead and then a reminder on the day. So if you have time in your schedule, please join. So some more exciting Aster talks coming up too.